So the signal transduction pathways starts off with reception. So a ligand, and in this case epinephrine, attaches to the receptor on the G protein. This turns on the G protein, which means a GTP replaces a GDP. Then the transduction starts, so the peripheral protein changes shape. So one of these detaches and comes over to the adenylyl cyclase. This signals for the adenylyl cyclase to capture an ATP and produce CAMP.
Okay, and then once the CAMP is produced, which a lot of CAMP is produced, it'll come over to the protein kinase, which is being inhibited, and it'll strip the inhibitor off the protein kinase. The protein kinase is then able to come over to this ATP right here and take the phosphorus off of it. A phosphorus, yeah, and which will leave an ADP rather than an ATP. Then the protein kinase will bring the phosphorus over to the enzyme, which in this case is phosphorylase, and that will turn it on, which then allows it to phosphorylate glycogen, which releases glucose and comes over here. And then when the glucose is produced, it allows for cellular respiration.
So the way Bacterium cholera inhibits this entire process is that the bacterium doesn't allow for the replacement of GTP with GDP. So that's the use of GTPase is the use of GTPase, the GTPase allows for the replacement of GTP with GDP, which turns off the G protein. So if this, so the GTPase is inhibited, so CAMP is continuously produced in large quantity.
And when the CAMP is continuously produced, it'll, in this case, instead of cutting up glycogen to produce glucose, it'll be producing large amounts of sodium and chloride and other things such as electrolytes. And all of these things will flood out of the cell. So outside of the cell, there will be a high concentration of the sodium chloride and electrolytes, etc. And then since the outside of the cell is hypertonic compared to the inside of the cell, the water from inside the cell will flood out, causing extreme dehydration. The end.
With only 3
guest
interruptions.
stars.